Okay, so today we're starting to talk about sensation and perception. And so there's going to be some repetition from what we talked about last time with the nervous system. Can anybody name the senses in the body? How many are there? 5. Okay, 5. What are they? Taste, vision, smell, touch, hearing. Okay. But we've also forgotten equilibrium, okay, knowing whether you're right side up or upside down. And also don't forget proprioception. What was that? Yeah, knowing where a body limb is relative to the rest of your body without having to watch it. So proprioception. So a stimulus is any type of sensory input that the body can have. And the receptors are designed to detect that stimulus and convert it into nerve impulses. Because basically, all this information is going where? The brain to the spinal cord. And the nervous system only understands nerve impulses. Like, yeah, you might be seeing light right now, but that's because your eyes are taking that pattern of light and converting it into nerve impulses that your brain understands. And so whatever a receptor does, it has to convert a stimulus into a pattern of nerve impulses. Okay, so we divide the senses up into the general senses and the special senses. General senses are senses that are distributed all over the body. For example, pain. If I were to hit you in the thumb with a hammer, would that hurt? Yeah, what about the forehead? Yeah. You have the sense of pain all over your body. We also call that nociception. Temperature, the same way. Your skin can detect hot and cold. And then touch and pressure. We have touch receptors all over the body. Now, in humans, they're more densely organized on the fingertips, but we still can detect touch, in the armpit or on the back or things like that. And then proprioception is located all over the body too. In all the different joints, we know when they're moving and what they're doing because we have a sense of proprioception. On the other hand, special senses are located in the head. That's because mammals tend to be cephalized. All our special senses, such as taste, smell, vision, hearing, and balance, are located up here in the head. Okay, so the types of receptors. The receptors are basically evolved to detect certain type of stimuli. And we can classify these receptors based on the type of stimuli they detect. So mechanoreceptors detect mechanical stimulation. Okay. Give me an example of a mechanoreceptor in the body, or a sense that uses mechanoreceptors. How about touch? Yes, touch is an excellent example of a sense that uses mechanoreception. When we poke you in the arm, okay, special touch receptors transduce that touch into graded potentials and action potentials. But hearing is also a mechanical receptor. The hair cells in your ears detect sound and convert that into nerve impulses. Thermoreceptors detect temperature. We have them in the skin, but also in areas of the brain that detect body temperature. Okay. And nociceptors detect pain. They're a type of mechanoreceptor sometimes, or even a chemoreceptor. And then photoreceptors detect what? Light. Okay, so the retina in the eye would be an example of a photoreceptor. And chemoreceptors detect what? Chemicals. Okay, so where in the body could we find a chemoreceptor? Yeah, that would be an interoceptor. Let's go think more common than pituitary. What's another example of a chemoreceptor? Taste, right? Taste and smell. Think about your favorite foods and why you like them. Okay? What's your favorite food? Somebody tell me. Pasta. Okay. And you like it because it has a certain taste, certain mouthfeel. But in reality, you like it because it's a combination of all these different molecules that are detected by the taste buds on your tongue, as well as the receptors within your nasal passages. So they all deal with chemicals. Okay, and then we have osmoreceptors. What do they detect? What word does that sound like? Osmosis. So these help detect water and salt balance. Okay, osmoreceptors tell your body when you're thirsty. Okay, when you're dehydrated, and they tell you, oh, I want to drink something, I'm thirsty. Okay, so sensory receptors look very different depending on the type of stimulus they're designed to detect. If we look at photoreceptors, these would be our rods and cones. They look like this, and again, the color is just artist rendition here. Whereas hearing receptors are really mechanoreceptors. These are the hair cells we'd find in the ear, and these would be the smell detecting cells that we see in the nasal passages. And similar cells we also find in the olfactory epithelium. Now, when we look at touch and pain, we either have an encapsulated receptor or a non-encapsulated receptor. So encapsulated receptors like Meissner's corpuscles, Pacinian corpuscles help us to detect touch and pressure. Whereas these unencapsulated receptors or free nerve endings detect things like tickle and itch and also pain. Okay, I know last time we talked about the process of transduction and transmission, but I would like to go over it again in context with pain. So let's look at somebody that's poking their finger with a needle, and that's stimulation, but we have to be able to convert that stimulation into a graded potential through transduction. So what kind of receptor would be responsible for transducing this type of stimulus into a graded potential? What kind of receptor? A nociceptor, which in this case might be a type of mechanoreceptor. It's a free nerve ending. So we stimulate that free nerve ending, we get transduction, we get basically a graded potential. Now, a graded potential is different than an action potential in that it's limited to the cell body and the dendrites, and that if we don't get enough graded potentials, they'll die off without having an action potential. But if we get enough graded potentials, then bam, that same neuron does transmission. Transmission is generation of those nerve impulses and the propagation across that axon until we get to the spinal cord. Okay? Now within the spinal cord and even the brain, we can have modulation. We can amplify that signal or dampen that signal. Okay? That's one thing that the central nervous system does. And eventually that information is going to be transmitted to the brain where we're going to be conscious or perceive that. So the cerebral cortex is the top part of the brain, right? Okay. We can actually become aware of a stimulus, particularly pain, maybe within the thalamus. But it's only on the cortex that we're able to say, not only, oh, I'm experiencing pain, but it's coming from my right arm or something like that. So it helps with localization. You've heard of anesthetics and analgesics. These are things we can have to prevent or treat pain. And anesthesia, the definition here is loss of sensation. And so anesthetics can be classified as local anesthetics or general anesthetics. General anesthetics are the ones that knock you out. They render you unconscious. Examples here would be isoflurane, which is our gas anesthetic, propofol, and even ketamine. Local anesthetics temporarily numb a certain area of the body. For example, lidocaine, bupivacaine are local anesthetics. Why would we use a local? Give me an example of a procedure we might use a local on.
Like stitches?
Stitches. If you're giving stitches, sure. What else? What else if you had a cane?
Dental work?
Dental work. You're probably going to have lidocaine or bupivacaine or other types of local anesthetics. And then we have analgesics. So analgesics reduce the perception of pain in various ways. And probably the most common analgesics you have in your home would be NSAIDs. Okay? These are non-steroidal anti-inflammatory drugs. Okay? These are drugs going to be like aspirin, ibuprofen. Okay? In the veterinary world, the common NSAIDs are like Rimadyl and meloxicam. And we have other drugs that are also pain reducers, such as the opioids. Okay? Opioids are drugs that are similar to morphine. So give me an example of human drugs that are opioids. Opium, yes, if you just have that in your opium den in IAEA or whatever, okay. Starting to get a bad impression of this place. Yeah. What else? Hydrocodone, oxycodone, all the codones. When you talk about Vicodin, right? Okay. These are all brand and generic names of opioid drugs. And then in the veterinary world, we have something called tramadol, which is sort of like a synthetic opioid as well. The point I should make is that not all anesthetics are analgesics. Many of them will render an animal unconscious, but actually have no effect on the process, the pain process. So let me give you a scenario here. We've brought a 6-year-old cat in for her first dental procedure. Never had a dental prophy in her life, but has been experiencing some pain, some odor coming from the mouth, inflammation, stomatitis. And so the veterinarian says, sure, bring her in. Let's do a dental prophy. Now, if it's been 6 years, this animal's never had a teeth cleaning, what will this prophy involve, do you think? What do we do during a prophy? Dental prophylaxis. Clean the teeth, okay? But part of a veterinary prophy can also be assessing the mobility of those teeth. If this is an older cat, it might have some mobile teeth, some teeth that actually need to be extracted. So, knowing that in mind, we want to prepare for the procedure, and usually we're going to say, well, we need to have some pain medication on board. But if you go to some clinics, they can just gas that cat down with isoflurane, which is an anesthetic, a general anesthetic, render it unconscious and do the prophy, okay, do the cleaning. But if they discovered that they needed to pull teeth, they don't really have any pain medication on board. And so even though the animal's unconscious while they're pulling the teeth, when it wakes back up, it's going to be in a lot of pain. And this is something we call wind-up. Wind-up happens because even though the animal is unconscious through general anesthesia, the pain pathway is still being stimulated. We're still having transduction of these pain impulses and transmission to the spinal cord. And then those are eventually going to be sent up to the brain. And the brain will become aware of these when we wake back up that animal and it has no pain medication on board. So we can even get so much pain that that animal can experience something called allodynia, which is where stimuli that would normally not be painful are perceived as pain. You've got a really painful cat, you go to touch it, it might yell out in pain, even though that stimulation might normally not be painful. People that have migraine headaches sometimes experience this. Anyone have migraines? Okay, when you look at the light or something like that and you're like, oh, light, oh, Jesus, you know. So allodynia is common when the sensory neurons are just overloaded, the brain's being bombarded by these pain impulses to where pretty much everything coming in is interpreted as pain, which is bad. Now, preventing wind-up is pretty easy, actually. In order to prevent wind-up, we need to use both preemptive and multimodal analgesia. Okay, what does preemptive mean here? Beforehand. So before we have that animal in surgery or start doing extractions, we should get the pain meds on beforehand. You know, and here we're talking about butorphanol, buprenorphine, even morphine. Morphine is very cheap, and they use it a lot in shelter medicine. So if you get that drug on board beforehand, you're going to actually reduce that animal's pain state. It's not going to experience near as much pain. The other thing is, because you've got the drug on board before the pain starts, you'll actually use a lot less of it, okay, which is important. And also multimodal analgesia. We want to break up the pain pathway at multiple spots. Okay, we want to prevent transduction. We want to prevent transmission. Okay, and so this is how these different analgesics work, and we talked about them briefly last time. Okay, things like our NSAIDs, like Rimadyl, meloxicam, they prevent inflammation and thereby reduce the amount of transduction going on at the terminal nerve endings. Your local anesthetics, your bupivacaine, your Americaine, they reduce and stop the nerve impulses on the afferent sensory neurons. Okay. And then we talk about drugs like our general anesthetics. Well, they prevent the perception of pain, while our opioid analgesics modify or modulate that pain. So if we're doing things right in the modern veterinary clinic, your veterinarian will use a cocktail of 2 or 3 different drugs in order to stop the pain pathway at several spots. And in doing this, you're going to get the best outcome in an animal that comes out with a minimal amount of pain. And remember, pain is bad, and because pain also will prolong healing time. Okay? Pain normally has a protective function, but too much pain is bad because it prolongs healing and causes other problems as well. Okay, when we go back to sensation and perception, the sensory receptors can be simple or they can be quite complex. Okay? When we talked about pain receptors, they're relatively simple, one-cell models where the same cell does the transduction, creating the graded potentials, as does the transmission, creating the action potentials. But we can also have a multi-cell model like in the retina, where one cell does the transduction, creates neurotransmitter, and then sends that neurotransmitter to another cell which actually does transmission. Now, there's something weird called receptor adaptation. Okay? Certain receptors, after they're stimulated repeatedly, will eventually stop reporting in. Okay? An example here is think about the touch receptors in your own skin. And when you move around, you're aware that you're wearing clothes, right? But this touch receptor, once you put your clothes on, or like you're wearing clothes, you're wearing clothes, you're wearing clothes, oh, what the hell, okay? It stops reporting in until you move again and it starts reporting in again. So it's a way for the body to filter the stimulus and only concentrate on really important things. And those are the things that are usually changing. Other examples here are the sense of smell, okay? If you ever walked in a room where somebody's wearing a lot of perfume and you're like initially going, oh my God, that's just way too much. But after a few minutes, you accommodate to it. It doesn't bother you. So dogs and cats do accommodate as well. We don't know it so much with smell, but we do know that they accommodate to, for example, the sense of their collar being on their neck. They're initially not aware of it unless you're tugging on it. Okay? So a lot of the senses do accommodate. One that does not accommodate is pain. Okay? When you're feeling pain, it doesn't usually go away until whatever's causing that pain is repaired. Okay, taste. Okay, what kind of sense is taste? What are we detecting here? Chemicals. Okay, so these are chemoreceptors. And I want you to think in terms of evolution. Okay, these chemoreceptors are really important for the animal's survival. Why?
They don't eat rotten flesh.
Yeah, so they don't eat something that's going to make them sick. Okay. My dog doesn't seem to think that rotten flesh makes him sick, so he continues to eat things. But does anybody know the tastes that we have described in humans? The 5 tastes? Sweet, sour, okay, umami, bitter. Okay, savory is umami. What are we missing? Sweet, sour, bitter, salty. Okay. So these are the tastes we've been able to detect in humans. It's hard to test a dog or a cat to see if they taste the same things or different things. But we do know that dogs have a preference, right, for umami foods. Okay. Umami foods are foods that are high in amino acids. They're the savory foods. Okay. MSG, monosodium glutamate, is basically a synthetic umami molecule. Okay. Now, why would it be to the advantage of dogs and cats to have a preference for things that have umami taste to them? So they know it's meat, right? So they know it's high in amino acids, probably has some fat along with it. Okay. And for cats, it's particularly important because, remember, they cannot be herbivores. They have to eat meat to get taurine. So it's important to their survival that they be able to say, yes, this is meat, I need to eat that. Okay, what about sweet? Do pets have any preference for sweet things?
Ice cream.
Ice cream, okay. And what did you say?
Bread would that be considered sweet?
Yeah, bread's not really sweet, it's starchy.
Strawberries.
Yeah, my dog loves sweet things, okay, fruits and things like that. Sugar, I'll eat it. It's not good for him. But why would it make sense that animals, including humans and dogs, have evolved a preference for sweet things? What does that tell us? Tells us they've got simple sugars in there, easily digestible and absorbed. Okay? Again, cats hopefully not so much, but they do like sweet things. And this sometimes can work against us. Think about things that are sweet that cats and dogs shouldn't be eating. Antifreeze, okay, chocolate, okay. So antifreeze is sweet. In fact, some people use that to poison cats and they'll go and lick it because it's sweet. Okay, what about sour? Now, I don't know that animals have a preference for sour, at least not dogs and cats, but what kind of foods that you eat are sour? Lemons, okay, limes, oranges, citrus fruit. And so initially you might get a craving every so often for sour things. Sour things tend to be higher in vitamin C. So that tells the body, okay, this is good, I might be getting some vitamin C. Whereas bitter things, what kinds of things are bitter? Bitter melon. Everyone gets me that in Hawaii. Coffee. Okay. A lot of plants are bitter. A lot of plant alkaloids, a lot of toxins are bitter. And so initially, think about young kids. Do they like bitter things? No. Kids have an aversion for bitter. Bitter is an acquired taste. And certainly animals do too. If you give your dog something that's bitter, they're going to eat it. And by avoiding bitter foods, they might be avoiding some phytotoxins, some plant toxins that are in the environment. So the structures in the mouth that are responsible for taste are papillae. These are the papillae here. Papilla just means bump. And remember, we have some pointy papilla on the cat's tongue. These are called filiform papilla. And we also have some more mushroom-shaped papilla called fungiform papilla. So the fungiform papilla are the ones with the taste buds. The taste buds are in the side there, and then surrounding that papilla, there is basically a cavity that would be filled with saliva. So this moat of saliva is around our papilla, and that's important because the saliva's job will be to dissolve the tastant molecules, the food molecules, so that they can be exposed to the taste buds. Okay, so here's our taste bud. We've got a couple different cells there. We've got our receptor cell, which is right here. We've got supporting cells, and we've got a basal cell. The basal cells can regenerate into new receptor cells, which is important because have you ever lost your sense of taste?
Yeah.
What happened?
Anesthesia.
You burned it and you said what?
Anesthesia.
Anesthesia, you lost your sense of taste? You had a cold? Okay. But certainly think about hot pizza or something like that, right? You can destroy these cells very quickly, but fortunately they do regenerate over time. So that's the job of the basal cells. Now, if we look at the physiology of gustation, we say that our dog has just gotten a hold of this hamburger here, and he's going to chew up the hamburgers through mastication. Those molecules will get dissolved in the saliva, and they'll bind to our gustatory hairs on our receptor cells. Okay. The receptor cells will then transduce that stimulus into a graded potential, and it will then release neurotransmitters which will connect with our afferent sensory neurons, our primary sensory neurons here. And those primary sensory neurons will be the cells that form the action potential and send that information to the central nervous system, where it'll say, hamburger. Okay? Now let's look at smell. So smell is also a different type of chemoreception, and it's important because smell and taste work very well together, right? As people get older, their sense of smell tends to go down a bit, and since their sense of taste goes down a bit as well. But dogs have a tremendous sense of smell, a much more developed olfactory epithelium that we have, and much more area of the brain dedicated to interpreting those smells, right? When your dog goes out and is sniffing things, it just for hours and hours can tell a lot about another animal by the scent of its urine and so forth. Okay, so here we have the same type of cells, our olfactory receptors, and then we also have basal cells that can regenerate those receptor cells and some supporting cells. Now the important thing is we don't have saliva here, but we do have mucus. So those molecules that we're breathing in are in the air, but then they dissolve in that mucus and bind to the olfactory receptors where they're going to cause a graded potential. Okay? And once that graded potential gets great enough, it will cause something called a generator potential. This is just an action potential that happens in the same cell. So remember I talked about a 1-cell and a 2-cell model, and when we talked about taste buds, that involved 2 different cells. But when we were talking about the cells within the olfactory epithelium, the single cell that does the transduction also does the transmission. And that action potential will then be propagated up towards secondary neurons, which will then carry that information into the central nervous system via our olfactory bulb or olfactory nerve. Okay, now let's go look at the ear. Now the ear is important for 2 different senses, hearing, and what is the second one? Balance and equilibrium. Okay, so let's take a look at the parts of the ear. So the ear is divided into 3 parts: the external ear, the middle ear, and the inner ear. The external ear is basically a funnel for sound, right? Okay. The earlobe here is called the auricle or the pinna, and its job is to funnel the sound, into the middle ear so that sound can be amplified. So if you look at the external ear, we have two parts here. We have a vertical canal and then a horizontal canal. What do people have? Just a horizontal canal, right? Okay. And then at the junction between the horizontal and the vertical canal, then after the horizontal canal, we have our middle ear. So the middle ear is separated from the outer ear by the eardrum or tympanic membrane. So this is when you're digging in your ear with a Q-tip and you hit something and it hurts. That's your tympanic membrane. And it's a very thin sheet of tissue that vibrates as sound waves come in, and then it activates these bones within the middle ear, which eventually push on this apparatus within the inner ear called the cochlea. And the cochlea is where transduction and transmission occurs. Okay, so we're going to go through the process of how sound waves are converted into action potentials. Anyway, going back to hearing, before we go on to the process of transducing sound waves into action potentials, we need to talk about a disorder of the outer ear of dogs called an oral hematoma. Now, what is a hematoma? It's like a bruise. It's a collection of blood underneath the skin. Okay, hema meaning blood. And here it's called oral, A-U-R-A-L, referring to the ear, not oral, O-R-A-L, referring to the mouth. And so this occurs in dogs as a result, oftentimes, of scratching at the ear or having an ear infection, especially in the big floppy-eared dogs. They do that thing all the time when their ears itch. And the flapping back and forth opens up blood vessels in the ear, and they bleed into the ear itself, and it causes them to puff up like this. And so how would you treat this? Yeah, slice it open, drain it, and then we can't just leave it, because oftentimes it'll fill back up. They'll do what they call a mattress suture or button suture in there to hold the outside skin against the cartilage and everything like that, so it doesn't open up again and form another oral hematoma. At the same time, you want to say, what's causing this dog to itch so much that it's getting this hematoma? So does it have, otitis externa? Does it have something else? Does it have yeast? What do we need to treat? Otherwise, the dog will very likely get it again after the sutures are removed. Okay, so let's go through the process of hearing. We had our sound coming in from the auricle through the horizontal canal, and then it bounces into the tympanic membrane. The tympanic membrane is going to vibrate, and that's going to set up vibrations within the middle ear and the auditory ossicles. These are the malleus, the incus, and the stapes. And so as this membrane vibrates, they vibrate. It pushes on this oval window, which is a soft part of our cochlea, and the cochlea here is filled with fluid. And as this oval window is pushed upon by the stapes, it sets up a flow of fluid through here that will deform these two canals going through there. Now, before we go to the next slide, I want to point out something right here called the eustachian tube. What does the eustachian tube do? So it connects the middle ear to the oral cavity. Why is that necessary? Why is it important? Pressure. It helps to equalize pressure. So imagine you're in your car, you're driving over the poly. What's happening to atmospheric pressure as you go up? It's going down. But the pressure in your ear, the air in there, came from good old sea level. So relative to the pressure that's around you, it begins to expand. So you feel your ears filling full, and eventually it goes, ee, and it squeaks out of there, and the air is equalized via the eustachian tubes. And so that's why the eustachian tubes are important to equalize pressure. Okay, so remember that we have these fluid-filled canals, and that the canals are vibrating as the oval window is being pushed upon by the stapes. And so as these canals vibrate, this is going to force these hair cells here into something called the tectorial membrane. And the tectorial membrane sits on top of the hair cells. The hair cells do the transduction of sound. And the more sound coming through here, the more sound waves, the more it's going to mash these hair cells into the tectorial membrane. Now, the interesting thing here is that the basilar membrane is like a diving board, and then it has a fat floppy end but then a narrow, stiff end. And so depending on the frequency of sound, if it's a low frequency sound, it's going to stimulate the wide floppy end, and that's going to touch against those hair cells there. Whereas if it's a high-pitched sound, it's going to mainly stimulate the stiff, thin area of the membrane, and that's going to stimulate a different row of hair cells. So this is going to help the animal to transduce differences between low-frequency sounds and high-frequency sounds. And in general, cats and dogs have a wider range of hearing, can hear into higher frequencies than what people can, right? They have a better sense of sound, better sense of hearing than we do. Okay. The big picture here is that these hair cells only transduce the stimulus. They create a graded potential and then secrete neurotransmitter, which then crosses across to these sensory neurons, and they're the ones that generate the action potentials. So it's a two-cell model. The other thing that's important is to realize that these hair cells can be damaged and they don't regenerate, right? If you've been to a rock concert and you're like jamming out and you come outside and what do you hear? You know, what do we call that? Ring. Okay, tinnitus. So that's tinnitus, ringing in the ears, and it's usually a sign of hearing damage, and usually a sign of permanent hearing damage. Basically, the hairs on here can actually be broken off the hair cells, and from what we know, we don't think they regrow. So have to be careful about that. Now, the ears are also important for equilibrium. The cochlea was a part of the inner ear that was concerned with sound reception. On the other hand, when we talk about the vestibule and the semicircular canals, these are important for interpretation of motion and of our orientation relative to gravity. So the vestibule is this area down here that is responsible for sensing our orientation relative to gravity. Are we right side up or upside down? Whereas our semicircular canals sense rotation and movement through space. Now let's take a look at the vestibule. We have hair cells here, just like what we did in the ear, except the hairs here are covered with a thick, gooey jelly that has something in it called otoliths. What does oto mean? Ear. What does lith mean? Stone. So these are ear stones. They're really heavy, and you know, mammals have really tiny microscopic ones. Fish actually have giant big ones. So if you imagine this hair cell is sitting there, and it's got this gooey, heavy mass that's full of rocks on there. And if it's sitting on there, it tells the animal, you know, if gravity's this way, it's going to mash down the hairs and tell the animal, okay, you're right side up. Whereas if it turns over a little bit, this is going to go up the opposite direction and tell the animal you're upside down, right? Give it an idea of orientation. So that's the point here of this otolithic membrane. It tells the animal what's going on. It also helps to tell the animal whether it's accelerating or decelerating. Because as the animal begins to accelerate, this stuff, because it's heavy, is going to lag behind and tell the animal, okay, I'm accelerating. Okay. Now, interesting about this is that all animals tend to have some kind of otolithic way to sense balance relative to gravity. We found out that crabs have it. I just found out even that jellyfish have it. Now, in crabs, it's a neat little experiment. They found out that they don't actually grow their own ear bones, but instead, during development, the larval crab picks up 2 pieces of sand and puts them in these little canals that eventually become its inner ear. And so, just like in the humans and like in dogs, instead of having a jelly-like mass there's just a grain of sand sitting on that hair cell, and that tells the crab, okay, I'm sitting right side up. And then some researchers figured out, like, let's raise some larval crabs. Instead of putting them on sand, let's raise them on iron filings, so just powdered iron. So they did that, and at a certain point in development, the crabs pick up these little pieces of iron, put them in there, and they work just fine as otoliths. The crabs had perfect orientation. And then the researchers came in and held a magnet above the crabs. What do you think happened?
They lost their orientation.
Yeah, so what's the ear stone going to do? It's going to go up. It's going to pull off the hairs. And what is the crab going to interpret that as? I must be upside down, so I'm going to turn right side up. Okay, so you turn it over and stuff like that. It's a neat little thing to see how these otolithic organs work in helping to maintain posture relative to gravity. Now, semicircular canals are different because here we're talking about rotation And so you can see that they're oriented at all different angles. 3 canals. They're filled with fluid, and they have hair cells in there and these swellings in the ends here. The swellings are called ampullae. And within the ampullae, our hair cells are embedded in a cupula. Now the cupula is another jelly-like membrane, but it doesn't have any rocks in it, no ear stones. But what happens is this animal rotates its head this way or that, fluid is going to rush through these canals and it's going to bend the cupula one way or another. When it bends the cupula this way or that way, it's going to cause nerve impulses to be fired off in the afferent sensory neuron. So it's going to give that animal an idea of rotation. Okay. Do dogs get car sick? Yeah. And they get car sick when they're moving and they're getting confused about the motion. They can't necessarily see outside. I mean, you've probably gotten motion sick at one time or another. One thing I'd like you to try in lab, we can't do it here because we don't have any twirly chairs. Get 3 people, 1 person sitting in a twirly chair, and just start spinning them around slowly, and then speed them up and speed them up and speed them. Do that for about 30 seconds and then stop them. Okay? And when you stop them, have them open their eyes, and you'll see their eyes are going for just a few seconds. And that's called nystagmus. And basically what happens when you're moving that person around, you set the fluid in this middle canal, moving in a circle, and it's telling the body, we're moving, let's track that movement with our eyes. And when you stop the person for a brief moment, that fluid continues to move in that circular canal. And so the body keeps trying to track the motion that's no longer happening. Okay? So that can result in some discombobulation in an animal that's in a car or something like that, getting motion sick. Okay, let's go to look at the eye, which is probably the most complex sensory organ in the body. Okay. The eye is made up of 3 different layers. The fibrous outer tunic made up of our sclera, the white of the eye, and the cornea up here. There's a middle tunic, which is made up of the choroid, which is a very vascular tissue. And then the inner tunic is made up of the retina. Okay. So here's our sclera again. That's part of the fibrous tunic. That's the white part of your eye. You cannot see out of that part. And then the cornea up here is an extension of that fibrous tunic. Its main function here is for refraction. And refraction is what? Bending. It helps to bend the light. There are 2 structures in the eyes that bend the light. The first one's the cornea, the next one's the lens. The cornea itself is not really adjustable, but the lens is. So these are the 2 structures that help you to focus or accommodate light onto the retina. Okay. The next internal layer is the choroid. Lots of blood vessels in there that are supplying the retina. And you'll notice the choroid is primarily black. Okay. Except you were looking at a sheep eye, so what else did you see in it? The bluish reflective thing, the tapetum lucidum. So in animals that are nighttime active, they have a little bit of reflective material on there, and that helps them to see better in low light conditions. Whereas in humans, we don't have a tapetum lucidum. And in fact, we have a really dark pigmented layer out there because we don't want light bouncing through a second time. So letting light bounce through a second time would give us maybe a greater ability to see in dark environments, but it might also reduce our visual acuity. Does anybody remember a time when cameras, they used to use this stuff, film? Do you remember this? Okay.
Yes.
Yes, so you would take a picture and then sometimes it wouldn't wind right and it would take another picture of the same frame. Would you ever have that double negative exposure or something like that? And you would see 2 images superimposed on one another. You can imagine that that's kind of like what would happen in the eye, at least that's what I imagine, when light bounces back a second time. Yes, it gives us a better ability to see in darkness, but as a predator animal that's active in day, it might reduce our visual acuity or ability to make out fine details. So by and large, diurnal animals don't have a tapetum lucidum, but things like cats and sheep and crocodiles definitely have one because it helps them see better in low light environments. Okay, next we have our iris. This is an extension of the vascular tunic or choroid. And what is its purpose? Yeah, constricts or dilates. So what does that help us do?
So much light at a time.
Yeah, it helps us to regulate the amount of light that's coming in the eye. You know, if you're sitting in a movie theater and it's really dark, they're probably dilated, right? And you walk outside and they're like, weak, you know, they're going to constrict. And so it helps regulate the light coming in the eye. And then the pupil is just the hole in the iris. Okay. And now our third layer there is the retina, also called the neural tunic. Okay, what are the receptor cells in the retina called? Rods and cones. Those are the primary cells responsible for transducing photons or light energy into electrical potentials. Now the retina, what did it look like when you dissected the eye? You may not, Tissue paper, like wet tissue paper. Very delicate, somewhat translucent, very, very delicate membrane. If you have an animal or if a person that's received a blow to the head, it can actually separate the retina from the choroid, which if not repaired can cause blindness in that area of the eye. And then, okay, the lens back here is another part of the eye that's used for refraction, but it's adjustable. There are muscles that pull on it that either can make it flat like a pancake or they can bulge it up and make it more football-like. So that's going to help the animal focus on things that are either far away or close to them. And then up in front of the lens, we have something called aqueous humor. Okay? And in back of the lens, we have something called vitreous humor. Why are these fluids here? What is their purpose? Keep the shape of the eyeball, okay? To maintain the shape of the eyeball, because the eyeball's ability to focus is determined also by its shape. Now, interestingly enough, there is a disorder of the humors of the eye, potentially the aqueous humor, where we get too much of that fluid and it builds up, and it causes an increased intraocular pressure. And in humans and in dogs and cats, this can cause glaucoma, which can cause damage to the neural tunic, the layer back here of the retina, that could eventually lead to blindness. And so you know when you go to the human doctor and they put that thing and they do the puff of air in your eye, okay, it's looking for intraocular pressure. We have a little digital thing that can do it in cats and dogs. You just have to numb up their cornea and you tap it a few times and get a good intraocular pressure. Because yeah, there's drugs we can give them to reduce that pressure. Okay, then we have our optic nerve coming out of the eye that goes to the brain. Now, this area in front of the optic nerve is called the optic disc, and that's important because it's a blind spot. Okay, there's actually no photoreceptors right there. But the animal's not aware of it and you're not aware of it. You kind of just feel in around it. Okay, so accommodation is the process of focusing, and focusing is going to involve the cornea and also the lens. The cornea is passive, it doesn't change shape, but the lens can change shape. So if you're looking at something that's very far away, okay, you're going to have a nice pancake-shaped lens. So the light comes in this way, and it kind of leaves the same way. It's not altered very much. But if you're looking at something that's very close up, you're going to have a lens that's more football-shaped, so that the light comes in, gets focused directly onto the focal point in the retina. Okay, and so this is called accommodation. Now, as you get older in people, the ability to accommodate changes. Okay, how does your grandfather read the newspaper? Out here, right? Okay, whereas a little kid can read a book like this, right? Okay. This lens gets less elastic with age. Now, when you did your eye dissection, you pulled the lens out, right? What did the lens feel like to you? Skittles. That's what somebody said. That's the first time in years of teaching this class that I've ever heard it compared to Skittles, but it's pretty good. Yeah. It's hard there because it's been fixed in formaldehyde, but it does harden a little bit with age. So now let's talk about the retina itself and how it transduces light into basically a graded potential. Now, I'm only going to give an overview because the way in which this happens is actually quite complex. But let's imagine that this dog is looking down at the ground at something interesting, and the light bouncing off that object is coming through the retina. It's going to go through first our ganglion cell layers, then our bipolar cell layers, and then finally over to our rods and cones. Now, at the rods and cones is where the actual transduction takes place. So the information proceeds from here to here. Now, what's the difference between a rod and a cone? What do they do differently? One sees color and one sees more shades of gray. So the cones are the ones that help us see color, and the rods see shades of gray. Now, do dogs have color vision, or are they just black and white? probably not as much color as we do. They do still have cones. And so maybe the color's a little washed out. Now, interestingly enough, some animals actually see in spectra that we can't even see in. Some can see in the UV. If we look at bumblebees and even some fish and stuff like that, it's pretty interesting. But as far as we know, dogs see like a washed out view of color. So the rods there are transducing black and white, and the cones are more color vision. Now, rods are good because they're good in low light levels. When you wake up in the morning, it's 5:30, you look around your room, and everything's shades of gray. Okay, you look at your dresser, what do the edges look like? Are they sharp or are they fuzzy? Fuzzy. Things are fuzzy because we don't have the acuity that we do with cones. But they are really good in really low light levels. Your cones aren't even working in that light. But you get brighter light, then you get your cones involved, then you can see really sharp detail. Okay, so that's the advantage of cones. Okay, anyway, what happens here, the rods and cones are stimulated, and they're going to talk to our bipolar cell layer, which is going to talk to our ganglion cell layer. And the ganglion cells are the ones that actually form the action potentials. And those action potentials will propagate down our optic nerve towards the brain. To finish up today, I just want to talk about comparisons in visual fields between different types of animals. Now look at prey animals like your, let's say, cows and horses. Okay, how are their eyes oriented on their head compared to us? Yeah, they're wide apart, right? So if this is a cow or horse, the eyes are there. And so its visual fields are rather large, right? They can see a long way around them. It looks like a velociraptor or something like that. Okay, he's got 2 ears on each side for some reason. And then look at something like a cat. Okay. Cats' eyes are close together because cats are devious, right? Okay, what would their visual field look like? It's narrower, right? So it's going to be like this. But there's also going to be a large degree of overlap. And here, now this is all kind of wasted to me, right? If you look at something in the room, like I'm looking at the camera, I close one eye, close the other eye, I can still see the camera. So there's a really wide overlap. We don't have near the range of vision that the prey animals have. We have a narrower range of vision. We have this overlap, but why is that advantageous to us? Depth perception, okay, which is important if you're a predator animal and you're trying to track down and catch a prey item, or you're trying to throw a basketball, something like that. Whereas in prey animals, they don't care about depth perception. It's better to have larger field of vision so that you can keep an eye out for the predator animals. I mean, even just look at the chicken there, okay, which is a bird, and then we look at the hawk. Chicken's eyes are, further apart marginally than on the hawk's eyes. They're a lot closer together. So it has to do with, what type of animal, are they predator or prey animals, depends on the eye placement. Somebody in the last class said, well, what about sharks? Their eyes are way over here. But, do sharks use their eyes much for hunting? No, they've got all these other senses that we don't have. They have electroreception, and they can sense the movement of water and things like that. So they're not as visual as you and I would be in hunting. Okay, so anyway, this is what I was just trying to show. This is a visual field for humans, and it would be very similar for predator animals such as cats and dogs. Widely overlapping visual fields, which allows them to have greater depth perception in pursuing a prey item. Now, there's a few things we did not get to in lecture, and I would like you to read about them in your book. Please read about all the external structures of the eye, the eyelids, the palpebrae, the conjunctiva, the nictitating membranes, the tarsal glands, and then there's a couple disorders that go along with this. So please read about this. I won't talk about it in class, but it will be on your quizzes and it will show up on your exam. Are there any questions? Okay.